Hereby I open this academic ceremony at Maastricht University. My name is Ton de Goeie. I am the pro-rector of this ceremony and chair of the degree committee. First of all, I would like to welcome Mrs. Julia Nicole DeBenedictis, and she will defend in public her academic thesis, which is called Dietary Phytochemicals for Personalized Prevention, Navigating Complexities. Welcome all members of the degree committee, and in particular I would like to welcome the two supervisors, Professor Theo de Kok. He is Professor of Population-Based Toxicogenomics at Maastricht University, and Dr. Simone van Breda, and she is Assistant Professor of Nutrigenomics and Nutrigenetics. Welcome other members, but I will introduce the 6 opponents later during the ceremony, because otherwise you will forget all names. Welcome to the audience here in the aula, and I would like to welcome also all followers of the live stream. Mrs. DeBenedictis, may I invite you to give a summary of your thesis? The floor is yours, and I wish you success in the coming hour.
Thank you very much. Dear Pro-Rector, members of the assessment committee, colleagues, friends, and family, both here and online, thank you for being here today for the presentation of my doctoral thesis entitled Dietary Phytochemicals for Personalized Prevention, Navigating Complexities. I will first introduce the topic, then guide you briefly through my work. Rather than solely a matter of genetics or bad luck, the risk of developing many chronic diseases, such as heart disease, diabetes, Alzheimer's, and some cancers, is increasingly linked to our lifestyle choices. Among these choices, our diet stands out as a critical factor. Within our diet, insufficient fruit and vegetable intake has been repeatedly linked to chronic disease risk. However, most people don't consume sufficient fruits and vegetables. Vegetable insufficiency alone is estimated to contribute to an annual 27 million years lost to premature death or years lived with disability or chronic disease. One major reason fruits and vegetables are so beneficial is due to the phytochemicals they contain. Phytochemicals are bioactive compounds in plants that also contribute to their various colors. Every fruit and vegetable contains many phytochemicals, and some fruits and vegetables are known for having a large amount of a particular class, such as carrots being known for their carotenoid content. Approximately 20,000 phytochemicals have been identified, which are categorized into groups based on their chemical structure and study for their unique health benefits. Phytochemicals help our body in several ways. As antioxidants, they prevent damage to our body's cells and DNA by neutralizing harmful radicals and supporting the body's natural defense system. They've been shown to interrupt inflammatory processes often involved in chronic disease and induce enzymes that regulate blood vessel function and dilation. We can actually learn something about our systemic vascular health, including our risk for hypertension, heart disease, and diabetes, by measuring the width of the blood vessels in our eye, by just taking a simple photo. Phytochemicals also play a role in gene expression changes. They're known for inducing signaling pathways in the body by influencing what genes are being transcribed through epigenetic modifications, and by acting as and modulating transcription factors. But let's get back to some basics so we can understand this better. DNA is like a giant instruction book for our bodies. It contains all the information needed to build and maintain us. You can think of it like a bulky cookbook that you inherited from your grandparents. Genes are like individual recipes in that book. Each gene contains the instructions for making a protein that performs a particular function in the body. DNA is turned into RNA in a process called transcription. RNA is like a photocopy of a specific recipe from the DNA book. Imagine you want to prepare a dish from a recipe in your cookbook. You don't want to get this special book dirty, so you make a copy of the recipe. This way, you can take the photocopy to the kitchen and follow the instructions without damaging the original book. Transcription factors help turn specific genes on or off. In our cells, transcription factors are like the chefs who open the recipe book, select the appropriate recipes, and oversee the process of making copies to ensure the correct dish is made. The process of making a dish or protein from the copied recipe is called translation. Phytochemicals have been shown to influence this initial transcription step, impacting which recipes are being made. They've been shown to affect the transcription of genes involved in biological pathways like blood clotting, fat and sugar metabolism, and cell death, which are processes relevant to cardiovascular disease, metabolic syndrome, and cancer development. From person to person, our genes can have tiny differences called polymorphisms, which are like little variations in the DNA recipe book that make each person unique. Often variations don't change much, but some changes can affect how effective these proteins work in our body. When genes code for proteins related to oxidative stress, detoxification, and phytochemical metabolism, this can impact someone's susceptibility to developing chronic diseases or their responses to certain nutrients. An analogy is that someone's variant for a mixing protein might produce an electric mixer, while another's produces a whisk. Both function as mixers, but one person is much more efficient than mixing than another's. Genetic variability might help us understand why some people respond well to a nutrition intervention while others don't. Most studies on phytochemicals focus on individual compounds, which, while important, doesn't reflect the real-world context in which we eat food. Isolated compounds and supplements are also less effective at delivering a human impact as fruits and vegetables in their whole food form, likely due to the loss of complementary compounds affecting a wider range of biological targets. There have been some in vitro and human studies looking at the combinatory and potential synergistic effect of two compounds or foods at a time, such as polyphenol-rich food with a glucosinolate-rich food, or an anthocyanin-rich food with a carotenoid-rich food. The interactions between these compounds can potentially enhance or reduce their overall impact on health. So there's a need for human studies which look at more combinations of foods rich in different phytochemical classes and varying in complexity. My thesis aimed to embrace the inherent complexity of studying phytochemicals from fruits and vegetables for disease prevention by addressing four major themes. The first theme is complex mixtures and their synergistic effects. This was explored with the MyBlend study. In Chapter 3, an intervention, where we measured markers of chronic disease risk after fruit and vegetable intake alongside gene expression changes in healthy adults. Our second theme was food processing. We examined the impact of the initial preparation and storage of these foods on their phytochemical content and their subsequent health impact in Chapters 2 and 5. The third theme was to determine if genetic variability influenced who derived the greatest benefit from these foods, explored in Chapters 4 through 6. And our fourth theme was to evaluate if we can derive more meaning from transcriptional data by pulling out cell-specific responses from multicellular transcription data. My thesis mostly revolves around two nutrition intervention studies, the aforementioned MyBlend study and a previously conducted study called Phytome, where I applied innovative analysis techniques to produce novel findings. In this presentation, I'll highlight the results of the MyBlend study and only briefly mention the main results of the other chapters. The MyBlend study is a randomized crossover study where we investigated 7 different blends of fruits and vegetables. 4 blends of fruits and vegetables represent a particular class of phytochemicals, and the other 3 are combinations of the first 4, which increase in complexity. Participants followed the study for 7 weeks. They all had a 2-week run-in of a low fruit and vegetable intake and were randomized to receive 2 of the 7 blends for 2 weeks, separated by a 1-week washout phase. At test days, we measured phytochemical absorption, resistance to oxidative stress and DNA damage, changes in retinal microvasculature, or that eye vessel width I mentioned, and gene expression changes. We aimed, number 1, to determine how an increase in fruit and vegetable intake affects chronic disease risk markers and gene expression change in humans, what complex blend of fruits and vegetables promotes the largest improvement in these outcomes, and 3, will there be a synergistic effect from the more complex blends? 146 participants completed the study, with around 40 participants per intervention blend. We found that increasing fruit and vegetable intake significantly improved plasma antioxidant capacity, provided increased protection against DNA damage, and improved retinal microvasculature. Blends rich in carotenoids, flavonoids, and the most complex blend offered the greatest reduction in DNA damage susceptibility. Anthocyanin and carotenoid-rich interventions were particularly effective at boosting antioxidant capacity, whereas improved retinal microvasculature was most evident in groups consuming blends with high flavonoid content and the most complex blend of fruits and vegetables. And a synergistic improvement was found in those that had a mixture of both flavonoid and anthocyanin-rich fruits. Moreover, significant gene expression changes were observed. Interestingly, DNA repair pathways were underrepresented after all fruits and vegetable interventions, suggesting that the body did not need to devote much energy towards repairing DNA damage with the added antioxidant defenses from the diet. Also reflected in the increase in antioxidant capacity in participant plasma and the reduction in DNA damage within participant cells. In Chapter 4, we aim to determine the role of genetic variability on the effects of different fruit and vegetable blends. We identified and tested 15 relevant polymorphic genes. Let's zoom in on XRCC1 and GSTP1. XRCC1 is a gene that produces a protein that helps repair DNA damage, preventing mutations which, if uncorrected, could lead to cancer. GSTP1 is a detoxification enzyme which may contribute to an individual's response to environmental exposures and has been shown to protect against vessel endothelial dysfunction induced by environmental toxins and to interact with fiber byproducts to regulate pathways that are critical to atherogenesis. We found that people with a common variant of XRCC1 after eating flavonoid-rich fruits and vegetables had increased protection against DNA damage compared to other variants. People with a common variant of GSTP1 had a larger improvement in overall microvasculature diameter after the increase in fruits and vegetables compared to another variant. These results show that a person's genetics can modulate how much benefit they may derive from a particular blend of fruits and vegetables. In Chapters 2 and 5, we considered how food processing and storage conditions affect the phytochemical levels and subsequent health benefits. In Chapter 2, we compared the traditional high-heat method of pasteurization versus a high-pressure, low-heat method called pascalization, thinking that pascalization would better preserve phytochemicals than pasteurization in a complex fruit and vegetable mixture. However, each technique showed strengths for different phytochemical classes, so the ideal preservation technique depends on the phytochemical of interest. In Chapters 6 and 7, we explore themes 3 and 4 in the Phytome study. The Phytome study investigated the effects of eating processed meat products on colorectal cancer risk-related outcomes, like the formation of potentially carcinogenic compounds called N-nitroso compounds, DNA damage either measured by strand breaks and adducts, and gene expression changes in colon tissue. In this initial study, there was a decrease in these N-nitroso compounds, but otherwise no significant changes were seen when people ate processed red meat versus processed red meat with added plant extracts. In Chapter 6, we found that after an increase in processed red meat intake, variations in the gene NQ01 were associated with levels of N-nitroso compounds formed in the colon, and variations in the gene COMT were linked to the level of DNA damage. This suggests that some people might be more genetically susceptible to the carcinogenic effects of processed red meat than others. After the addition of phytochemicals to the meat, variations in GSTP1 related to how well the added plant extracts led to reduced DNA damage. We also implemented machine learning techniques to determine how the combinatory effect of all the measured genetic variants impacts someone's overall response to the addition of plant extracts to processed red meat. The combined gene score that we developed did well with correlating if someone responded to the diet, meaning if someone had an improvement in nitroso compounds or DNA damage. Also, gene expression changes were analyzed between the GSTP1 variant groups to identify potential mechanisms impacting the improved reduction in risk due to the differences in genotype. In the original Phytome study, the analysis of gene expression data from multicellular tissue showed no significant differences between the different diets. However, in Chapter 7, we used a bioinformatic algorithm to uncover cell-specific transcriptional changes, which produced a novel and relevant findings. Unlike a drug, which usually affects a few biological processes in a pronounced way, diet may affect many processes but to a more subtle degree, which can often be overlooked by typical analysis methods. So deriving cell-specific responses from multicellular tissue can reveal these subtle but important alterations. We found that adding phytochemicals and reducing nitrite in processed red meat increased stem cell and goblet cell transcripts while decreasing transcripts from transit amplifying cells. This, plus the changes in gene expression by cell type, suggests that adding plant extracts to processed red meat may aid with mucus layer restoration in the colon, enhancing its defense mechanisms. In summary, this research navigated the complex intersection between dietary phytochemicals and personalized prevention. Processing methods in functional food production should be tailored to the specific phytochemicals highlighted in a fruit and vegetable product. No single fruit and vegetable blend outperformed all others across all markers. Thus, our research underscores the importance of personalizing dietary recommendations based on an individual's health profile, environmental exposures, and disease risks. Grouping participants by relevant genetic polymorphisms and utilizing machine learning techniques showed who had the worst effects from processed red meat and the most benefit from eating a specific blend of fruits and vegetables. And finally, tools like CyberSort revealed diverse gene expression responses in colon tissue after different dietary interventions, enhancing our understanding of potential underlying mechanisms. Thank you for your attention. I will now pass the word to the prorector.
Thank you very much for this clear summary of your thesis. The opposition will be opened by the chair of the Thesis Assessment Committee, Professor Dick Seym. He is Professor of Food Safety in a Sustainable Economy at Maastricht University. Professor Seym.
Thank you, and dear candidate, first of all, I would like to congratulate with finishing such a beautiful book.
Thank you.
A lengthy book, and so much in it. And the complexity word in the title really means something. But I also must conclude that it is still not thick enough, because you missed the supplementary chapter in chapter 3. So I had to look it up, and I did. And because I had a question on that chapter. First of all, I noted that I understand that you only chose adults, because group number 4, I don't think any kid would consume that Brussels sprout and broccoli. On the vitamin C issue, you mentioned something that with increasing vitamin C levels, there was an increasing urine excretion. But why did you mention that so specifically? Because it makes sense. If you put in more, there will be more excretion. But what? And if I looked at the figure in the supplementary part, it didn't really tell me anything. So could you explain what you did and what it means?
Yes, highly esteemed opponent, thank you for your compliments and also your question. So you mentioned a couple things. One was, choosing adults. Some of the interventions were maybe even a struggle for some adults to consume in the way that they did. But specifically with the vitamin C measurement, we assume, of course, that when we give participants a dietary intervention, they go home and they consume the intervention. We want to do also some extra checks just to validate that, yes, indeed, when we expect there to be an increase in vitamin C, either intake or excretion, that we're actually seeing that, which gives us some validation that the participants did follow the study as directed, but then just gives us concrete data that we can then use for potential other analyses.
Okay, thank you. My next question is a much bigger one, and I actually would like to challenge your proposition number 9. So I would like to invite one of your paronyms to read out proposition number 9.
This thesis doesn't only confirm that an increasing fruit and vegetable intake reduces markers of chronic disease risk, it also laying the groundwork for personalized nutrition strategies by showing how genetic variations can influence the effectiveness of nutrition interventions.
Thank you. My observation is that in different parts of the thesis, you end up with there's more research needed, but there's also a groundwork for personalized nutrition strategies. My first question would be, how would you define person or personalized?
Very much for your question. So the personalized aspect of personalized nutrition, for example, similar to personalized medicine, but let's focus on nutrition. It stands in contrast to the approach of public health efforts. So public health efforts are very important for a population if we have general guidelines that people can follow. So you've probably heard some of them, and it kind of gives the general public an idea, okay, so what foods, if I'm concerned about my health, are going to be more ideal for me versus others. That's important to have. But when we are talking about personalized nutrition, the problem with public health in general, the one major pitfall is that you can give this overarching advice, but there will be people for whom that advice is not true. And so they may be, choosing certain food behaviors which you might expect an improvement, and they're not, due to potential differences in genetics. It could be differences in lifestyle. It could be differences in food tolerance.
Yeah.
Many reasons.
If I may interrupt you there.
Yes.
Now it seems like you feed people, fruits, vegetables, other stuff, and then it's taken up and you taken bioavailability into account, and then you assume that then the reaction or the follow-up will happen, the phytochemicals will act in the genetic schemes and so on. But how about the microbiome? Do you think it's part of the person? Because it can also be affected by the fruits and vegetables and the phytochemicals in it, and that can then consequently be affecting in an epigenetic way also the person.
Yes.
So how could you deal with that and in also personalized advice?
I completely agree with you that the microbiome plays a huge role in this interaction, because our gut microbiome really impacts our response to nutrients as well, and also how those nutrients are then converted, for example, to short-chain fatty acids, which are then reabsorbed in the body and have an additional impact. For example, polyphenols, when you consume them, you see also this increase in absorption when you expect polyphenols to be in the colon. So we assume that there's some action of the microbiota in the absorption and the availability of these nutrients in general. So the microbiota plays a huge role. It is, as you said, this thesis is quite large, and even without the supplementary information, which I would have loved to include, but then we must save some trees. It is a topic that I would love to include, and I think that, and I hope that this other field as well, the microbiome field, continues to develop in a way that we can better identify patterns.
What would you study if you would have money and time and would study the microbiome in this aspect?
So I first would like to see the kind of genus populations that exist in each person's microbiome. I would like to see fecal samples, for example, in order to measure this. And then also something that's important when we consider the microbiome is not just the different families that we have, because we haven't assumed these are beneficial, these ones more or less, these ones may be harmful, but what are the actual activities of those microbiome families, because they might be involved in bile acid formation or breakdown of fiber, for example. And we know that also the microbiome changes when you consume different foods. So if you increase your fruit and vegetable intake, we expect a change in them as well, which then changes more things. I would also be interested in measuring, for example, like I mentioned, those short-chain fatty acids that get reabsorbed, because they have also really meaningful impacts on our chronic disease risk. So there's lots to be to examine on that in that world, and it plays a very large role, especially in our metabolism of complex carbohydrates.
Okay, thank you very much. I think, considering the time, I need to give back the microphone to the prorector. Thank you very much.
Thank you for your questions.
Thank you, Professor Sam. The next opponent is Professor Ellen Kampman. She is Professor of Nutrition and Disease at Wageningen University and the Division of Human Nutrition. Professor Kampman.
Thank you very much. And have to open. First and foremost, the candidate, congratulations with all the work that you did. It's a huge achievement and a beautiful book. And I would like to congratulate your supervisory team at the same time. It looks and it reads really like a novel. I very much enjoyed it. It's very original, I think, very creative work. And I could also see your creativity in all the figures and the graphs, and also in the way you explain it now with the DNA recipe book. I will never forget that. And you're also very creative in the methods that you used. And I would like to talk with you first on the figure on page 67, which really puzzled me and also showed the complexity. Yeah, everybody has it? Okay. It's a bit hard to read for me, but the figure, I'm not sure what I'm looking at. You say it's a cross-sectional study, a crossover study, a crossover study. And with a crossover study, I think you start with blend A normally, and then there was a period, and the same individual then goes to blend B, and then you compare the response to the two blends, and that's a crossover. Now, when I look at that figure, I see something different. Could you explain very briefly how you did this?
Highly esteemed opponent, thank you for your compliments and your question. So in a perfect world, we could have had every participant consume every single blend that we had. But enrolling participants for, I don't know how long that would be, many months, to follow an intervention study, likely we would see some many dropouts. It isn't feasible. So what we did is we did crossover 2 of the 7 blends. So each participant consumed 2 blends separated by a washout period.
Okay, and when I look at the figure, I think, could it also be possible that some people got the same blend? Because it's sort of twice the same study, so it's a repetition of the study. So you randomize them first over 7 blends, and then wash out, and then you randomize them again. So it could be that they have the same blend.
So we ensured that in the randomization process that each participant consumed a different blend. So they were randomized just before their first intervention period, so that we had the interventions already thawing because they were all frozen prior to this moment. And so in the randomization procedure, we of course had every participant consuming a different blend. There was no repeated, and working directly with these participants, it would have been clear if there was any mistake like this, so it never happened.
Okay, great. And there, of course, a huge number of combinations that you can get in this way.
Yes.
About 5,000, I calculated, but it might not be completely true. But
so we did also ensure that the number of interventions was balanced as well from the intervention phase 1 and intervention phase 2. So that we don't have, potentially for blend number 5, 80% of them being in the second phase versus, 20 in the first phase. So it was balanced as well as much as possible to ensure that they were evenly matched. But also in our statistical methods, we included the word just escaped me, but an order effect check.
Yeah.
So we made sure that there wasn't an order effect, meaning the first intervention impacts the second one and vice versa.
Okay, well, I'm satisfied you really thought about this. You must have thought also very carefully about the fact that you have a short washout period, 1 week.
Yes.
And you base that on a previous intervention study. But I was thinking in that paper you say it could be 2 weeks, better be 2 weeks, but you do now 1 week, and it's based on a meat consumption intervention. Can we compare that? And also you're looking there at responses in the colon, not in the blood. So food and fruits and vegetables and blood versus meat and the colon. Isn't that different to compare? Now, how did you come up with this 1 week?
Yeah, it's correct. There are different tissues that we're using to interpret how we might expect this amount of time to serve as that buffer between interventions. So there is some inference being made, but given as well the gene expression changes that we saw, that there was sufficient time in that previous study, it seemed like an appropriate amount of time between the two, and we also double-checked this when we collected the data from the participants, including carotenoid absorption, which we do expect to last a bit longer. The levels in plasma, we did see that there wasn't this dose response, for example, after intervention phase 1 and 2, where we think like, okay, they're loading on more. There wasn't enough of a stop between the two. We saw what you'd expect, kind of this level, and it wasn't dependent on the phases that they were in. So with our data, we did see some confirmation that the 1 week washout was sufficient time to see the effects of the first intervention not impacting the effects of the second.
Okay, great. I'm glad to hear that. Then I would like to move to the table 3 on page 70. because my favorite substance in vegetables and fruits is dietary fiber. And I can also see that really increased impressively. Also, of course, because you really have an extreme change. I also wondered whether that could not influence your findings. But let's keep it to the fiber. So the fiber, indeed, as you already discussed with my predecessor, you already said it has to do with microbiome changes, etc., etc. Could the fiber not be responsible for everything you see?
That's a good question. So we do have this big increase in fiber from baseline to the intervention phases. And as a dietitian, we typically recommend a stepwise increase in fiber to avoid any potential gut issues, because your gut is a major part in metabolizing these products. So that is something that just is inherent for the intervention to be working properly. And so the thing is, though, with antioxidants, or I guess the phytochemicals in the foods, one reason why we know that the phytochemicals are playing a role and it's not just fiber is because we also have many studies that look at the individual isolated compounds and their impact, as well as extracts and their impact, which exclude the fiber. So we have a lot of historical data to show that phytochemicals.
And those were also in humans. Sorry to interrupt.
Some of them were in humans. Many of them are done in vitro because you are much able to control that environment.
But that works differently.
Yes, of course. And there do need to be more research done in humans, of course. I think so, but of course, in vitro studies gave us extra important information.
Yeah.
But what we wanted to do with this study as well is to create interventions that also have a translatable power, because it's one thing to say, oh, get these fruits and vegetables and juice them with your juicer, and because also fiber is important, it's really about the whole food, then we didn't want to exclude it.
Okay, that's my last question. I think, do I have time?
Yes.
One last question, because I have so many questions, but let's move to the last one because of time. And it's exactly what you said. So it should be translatable to something that we can use in the public, with the public. And you actually, do you suggest that we should provide people with these blends instead of vegetables and fruit? As a dietitian, do you really think that this is the alternative that we should provide, and then people might be able to adhere to the guidelines?
So speaking as a dietitian, the way that we administered the interventions was basically a smoothie format. They're blended and they're put into a bottle and they're consumed much quicker. So there are positives and negatives to recommending whole fruits and vegetables. This is maybe the ideal, what we hope people consume. But if people aren't able to do that necessarily, then there's a next good option, which could be, blended fruits and vegetables with a fiber intact in a drinkable product. But of course, there are limitations when you have a product which is very quickly consumed. This is something I also advised the participants, was to consume them slowly, don't drink them all at once. We actually divided them into 4 bottles so that they consume them throughout the day versus just having one large load. And then you expect, of course, there's an increase in glucose, an increase in insulin, and we're really providing a lot of benefit.
And what about the fact if we wouldn't chew anymore?
Yes.
What about our teeth in the long run and our satiety system?
Yes. Correct. The satiety system is a huge component.
You may give a short answer.
Okay. I love this question, though. So I'll say very quickly, what we think generally is if you're not using it, you lose it, right? So we want to be incorporating that, and chewing your food, taking your time with your food does impact your satiety. It impacts how well you digest your food and how well you absorb those nutrients. So yes.
Blends, yes or no?
Blends, yes, if whole foods aren't a good option. But whole foods, fruits and vegetables are my preference, yes.
Okay, we agree. Thank you very much. I would like to give the floor back to the prorector. Thank you.
Thank you, Professor Kampmann. The next opponent is Dr. Joachim Schneider. He has a PhD in plant pathology, and he is currently Chief Strategy Officer at Antibodies in Germany. Dr. Schneider.
Yeah, thank you. dear candidate. As you said already, a beautiful book, very comprehensive. The topic is very close to my heart when it comes to personalized nutrition. From my previous job as CEO of a vegetable breeding company, I know that obviously fruits and vegetables are very healthy. It goes a little bit already in the direction that you had, but not many people are consuming the right amounts. That's number one. Number two, I know from my position at Inner Buddies, where we translate stool and microbiome analysis into personalized nutritional advice, that behavior change is extremely important and relevant when you want to achieve an impact. So when you take all of this together and your analysis of the quite already creative compounds that were developed, the mixes, but also the pearls, I found this quite creative. What would be your idea for a product that is, first of all, relevant from a biological perspective, but secondly, also impactful from a behavior change perspective and a practicality perspective? And last not least, maybe also commercially doable.
Esteemed opponent, thank you for your question and your compliments. Okay, large question. So essentially, if I were to create a product that is just generally bringing health, I think is the idea, correct? It would be one where, and if it was doable, so a product, we're not talking about whole fruits and vegetables that you find in your produce section of your grocery store, we're talking about something commercially made. I would think of something, and maybe I'm a bit biased, but let me first mention that one thing that I don't typically like to advise is consuming even fruits and vegetables, carbohydrates on their own. Not that there's a problem with it, but if you can consume it with fats and protein, you do have a nice effect on your satiety, and you have a slower absorption of certain sugars. There's other components that happen when you have a more well-rounded meal. So if we're creating a snack food, for example, something that you have in between meals, or you see a lot of recently these meal replacement products, things that are important are, of course, the phytochemicals that you're getting. So you want to make sure that you're providing those and a mixture of fruits and vegetables. I would like to see even a fermented product. So if we add probiotics within that, so potentially like a yogurt of sorts, or something like that where there's also enough viscosity, so like we explained earlier, that you're not consuming the food too quickly, and that it takes time, maybe something spoonable. So I would be interested in making sure that we were balanced across the macronutrients, that we have those added phytochemicals, that there's fiber still intact, that there's prebiotics and probiotics also included to give food for the microbiota that you have, but then also add extra microbiota. And I think that is feasible as a commercial product, but it does require a lot of balancing acts.
Which sampling of humans would be the best to maximize the personalization or the degree of personalization from a nutritional perspective? Obviously, microbiome, I guess, would be one.
Please, again, a short answer.
Do you mean a sampling of participants, or
No,
do you mean just what kind of
body samples?
So, like you already mentioned, yes, and one thing that we did, which was really quick and easy and a predictor of cardiovascular disease, diabetes, and cancer, is the retinal images. So seeing these small changes in the arterioles and the venules, you don't have to spend a lot of time in the lab. There's software to measure it, and you can get that moment of data capture.
Okay, thank you.
Thank you.
Thank you, Dr. Schneider. The opposition will be continued by Professor Matti Wijnberg. She is Professor of Molecular Epidemiology of Cancer at Maastricht University. Professor Wijnberg.
Thank you. So let me start also by congratulating you with this wonderful book. Really impressive, a lot of work, very different types of things that you did. So very nice to see. And also congratulations to your promotion team. I'm a bit worried. I'm an epidemiologist, so I have to ask you questions, I think, also a little bit on my topic. I'm a little bit worried in some of the studies with regards to the power. And I actually, when I started reading, I was really, really happy because there was a nice little chapter within your first, chapter 3, the first study that you described, the dietary interventions, where you did a power calculation. So I thought that was really well done, and you reached those numbers in most of the groups. And then I was a bit surprised that in chapter 5, where you had a similar study, but then looked at the coated pearls or non-coated pearls, that these two groups were smaller. Could you explain to me why you decided to do that?
Yes. Highly esteemed opponent, thank you for your compliments and your question. It's a great question. This was maybe some of the impact of COVID can be seen here. So we had to make choices as far as what was feasible for getting this sufficient power for the participants. So the MyBlend study, which was more robust and the one that was really the core of my thesis work, that was prioritized, and then we aimed for as much as we could with the pearls, which ended up being exactly what we estimated would be, which was about 20. So this was just our effort to do that. We did the power calculation based on the gene expression data as well. I was expecting that we need more participants because of all the extra variability that we would see differences between the groups. So there was a slight assumption that because of the power calculation being specifically to a noisy variable, that with the other variables that we used for that chapter, that we would have sufficient power for those.
Okay. So you're not worried that it really influenced your results? That maybe you're missing things that could have been there?
It could have. I think that I would have preferred that we had 40 across the board for that chapter. It wasn't feasible in the time that we had, unfortunately.
Okay. Yeah, because that kind of goes on also when you go into the genetics. You're looking at these genotypes, and the groups get really, really small.
Yes.
And especially for some of the interesting findings that you have, that you also presented in your introduction, for one of those genes, the XRCC1. I noticed that the significant difference that you're concluding about is based on a difference in which one of the groups only has 3 participants, I estimated. What was it, 7% of 40 or something?
Yeah, it was a small number. I think we had minimum 5 participants per group in order to consider it a group. But you're right. We chose only polymorphisms which have a 20% prevalence rate in the population based on previously performed studies. And then you collect a sample of individuals and you expect them to be completely representative of whichever studies have been previously published showing this prevalence. And then in our case, that just wasn't true. Some participants were more likely to have a polymorphism than another, so we had a lower prevalence rate. So that meant we had a smaller group, and indeed, we saw in that smaller group this larger change.
Yeah, and maybe the larger numbers are due to that you combined the groups at the 2 phases. No, there was only one. There was only one.
That in the study where we look at the different genetic variability for the MyBlend study, we did combine some of the interventions which represent similar phytochemical classes in order to bolster that power.
Yeah. Okay. So I was interested in those genetics, and you also repeated a little bit what was observed previously, so that kind of, I think, helps in your discussion, even though it's your conclusion, even though it's based on very, very small numbers. I really like that in chapter 6 you started also using a risk score.
Correct. Yes.
And I think you did well on that, and also describing how you did that. And then I was kind of surprised that you looked at the individual genes again. And I was then later on surprised in your discussion that you didn't take it a little bit further for future studies. Do you think there's room for using GWAS to get a really powerful risk score? If you want to take this to personalized prevention, you need powerful risk scores, I think. What
Yes.
do you think?
So what we had available to us at the time was the different genetic variants that we measured in the population. So what we had gene expression data, which you could kind of backwards track and get some genetic information in a larger way. And then we had measured specifically these 10 to 15 genes which coded for relevant polymorphisms that might impact the nutrient absorption of the intervention and their disease risk. So, and making sure that they're also prevalent in the population. So we had that information at our disposal, and that's what we used to develop the polygenic risk score. And I agree with you that if we had more genes, because we are a collection of many, and similarly what I said earlier is that we expect diet to impact a larger number of different gene expression changes, and there to be more wide-ranging approach versus, for example, maybe there's a simpler mechanism happening or a simpler biological pathway with a drug, for example. So we were limited in the risk score that we created on the genes that we identified, but this was the realm which we could operate in. So it's the data we had and it's the data we used.
So there are no GWASs available for the specific molecular outcomes that you investigated here?
There potentially could be. There aren't any.
Because I thought maybe that's the problem. I mean, it's not my area of expertise. That's why they're just not there.
Mm-hmm. No, it's more that we had other techniques available, like machine learning techniques, which could help identify from the gene set that we collected which ones are making the largest impact on the markers. And because we had these tools in our toolkit, this is why we choose that approach.
And I have to stop now, so thank you very much for your answer.
Thank you for your questions.
I'm giving the word back to the pro-rector.
Thank you, Professor Weyenberg. The next opponent is Dr. Sabine Langy. She is assistant professor in genotoxicology, nutrigenetics, and molecular biology at Maastricht University.
Thank you. Dear candidate, it was a pleasure for me to accept to be here in your jury. They only asked me a few weeks ago, but I read your thesis with great interest, and I also watch you do most of the comment analysis quite up close. So, of course, I'm going to ask a question about that. So my first question is mainly like, why did you choose the challenge assay to look at DNA damage or DNA damage protection and not any other assay? And also I saw in your methods that you say you also had unexposed cells. Were these control cells, or was it also blood that was unexposed? And if so, why don't we see standard breaks in the data?
Esteemed opponent, thank you for your compliments and your question. So the first question was, can you repeat it for me?
The first question was, why the challenge assay?
Why the challenge assay?
Yeah.
So there are other assays. There's repair assays, for example, which would be interesting, especially with our findings in the gene expression data, to be able to look at that. But when we sat down and looked at the sheer volume of work that had to be done with the COMET assay, and as you're aware, it takes a long time to perform this particular experiment. So we had about 150 participants and 3 time points, which is 450 or so samples, and then each sample was done in triplicate, and we had an unexposed and exposed. So it just became a mountain of work. And if we want to do it properly, we need to choose one. So, because otherwise you may get really interesting data, but if we spread myself too thin, then how reliable can we feel about the data? So we chose this assay. It's also one that in our department there was good experience in, so this is what we went forward with. And because we also want to, the interesting thing about the COMET assay is that in healthy adults, when we give them something like this, a lifestyle intervention, it can be a little bit more difficult to see significant changes in a short period of time. Mm-hmm. So when we have a challenge assay where we can take those cells, remove them from the body, and expose them to enough DNA damage or oxidative stress in order to see some kind of response, but also not kill the cells, then that's ideal, because we want to see in maybe like a over time or real-world situation, if they had a large exposure to oxidative stress, would they be able to withstand it? So that was the decision for this one.
Yeah, but it doesn't tell you anything about DNA oxidation as such, and not the levels they had, more the protection,
No.
so more like the antioxidant defenses.
Correct,
I would
and
say.
we measured the antioxidant capacity in the plasma as
Yeah.
kind of another way to see, okay, we see this increase in antioxidant capacity. We also see that their DNA is less damaged after the same exposure. And to answer your other question, I took the cells from the participants, the lymphocytes, which contain DNA, and I had carried those through the experiment, some cells which were not exposed and some which were. So we weren't doing exposed or unexposed blood, it was to the cell level.
But you didn't put the unexposed in the comment?
No, they were on the same slide. So we have an unexposed sample and an exposed sample on a slide, and then we had that in triplicate for each time point.
Okay, because the unexposed ones could have given you the strand breaks, like the standard background
yeah, that's
damage
true.
that people have.
Yes.
So I was a bit sad not to see these data.
The data,
in
yes, I see what you're
the-
saying.
Yeah,
Yes,
yeah,
I completely agree.
Yeah.
It gives you a sense of how much, in general, you would expect
Yeah.
on a basic level.
Level,
Yeah,
yeah.
good point.
Okay, then maybe to link it to the other marker, you said there's one other marker of DNA oxidation that you studied, which was in the urine, and although you didn't see significant differences there was a slight increase, I would say, in the aoxo in the urine. Also, I saw that there was a difference between females and males, which was large. So I'm not fully agreeing with you that it is a marker of DNA oxidation. I mean, where can you maybe elaborate? Where do these 8-oxo-DGs come from in the urine?
So my understanding is that it's more of a measure of your systemic, DNA damage. We take these samples from their blood and expose them, but that's just the lymphocytes that happen to be flowing there. But then when you have this, collection of oxidized purines from the DNA that then end up in the urine, you're getting more from an idea from the rest of the tissues. The level of the damage you assume is coming from that. It could be from just normal recycling of nucleic acids, for example. So yeah, that's where I expected they come from.
Yeah, a part comes from indeed turnover of nucleotides, but also from repair. So if you think in that way, I would say it also shows that you have an increase in DNA repair activity. Do you agree with that, or you say,
And this one, we did do this one across all of the
Yeah, you did
groups.
it for one blend, You did it
For one blend.
Yeah.
This one was a bit difficult to determine, really, because we just looked in one blend. And again, with the men and women, I think in this blend, we even divide that group even smaller. So, yeah, I looked in the literature as well on this particular marker, and I had trouble finding it being used in a similar context when you have healthy participants and having a lifestyle intervention like this, whereas it seemed more robust as a tool when you have people who have higher levels of oxidative stress generally in the body and are seeing changes that way. So my interpretation of this marker is that I don't think it's ideal for a study like this, but we took it just because we did want to see if there was, we want to have an extra marker for the same kind of outcome.
But yeah, now it's a bit contradicting because it could indeed show higher repair because higher cutting out of the 8-oxo-DGs. But on the other hand, you see gene expression going down, so repair going down.
Yeah.
So
Could be better turnover,
which
potentially.
would be
I have to
better-
interrupt your discussion. We
Okay.
have to move on to the next opponent.
Okay, thank you.
Thanks for your question.
Thank you, Dr. Longy. The next opponent and the last opponent is Dr. Susan Steinbusch-Kort, and she's Associate Professor of Systems Biology and Nutrigenomics at Maastricht University.
Thank you. Dear candidate, of course, I also want to congratulate you with an impressive amount of work. I think your book is both beautiful from the outside and the inside, so well done. And I also want to congratulate your supervisory team. I have some questions about chapter 7, about the cybersort X algorithm that you applied. I think you elegantly showed that there's a way to, from bulk transcriptomic data, get insight into specific cell types. You have colon tissue, and you designed a novel signature matrix for that tissue. How do you know that's accurate and it has all the needed cell types in that matrix? So maybe you can explain that a bit to me.
Esteemed opponent, thank you for your compliments and your question. So when we were looking for single cell RNA data that already existed because we didn't have that presently for ourselves. And we found one input into GEO of 3 healthy participants and their epithelium. So we downloaded this, and then we used a number of different methods we actually reanalyzed the data from the raw data through a pipeline, and we checked using, trying to remember the name, I think site type, to be able to identify the clusters first that existed in the different cell types, or what we'd expect to be different cells, and then we made sure that those clusters were clustering properly to understand, okay, these are different cells, but then we also double-checked that there were cell markers that we would expect from those cells particularly present in that cluster. So we did this kind of back and forth validation.
Yeah, because that site type was the gastrointestinal tract, right? The complete one.
Yes. And then there was also data from the original study where they also typed the cells and showed what cells should be expected. So there was this kind of cross-referencing.
Okay. And then you, in figure 1C on page 208, you nicely show in the different cell types the cell proportions depending on the time point that you have. So you have this intervention study, which you nicely explained in your introduction, where you have the processed red meat and the phyto meat, and you have now shown in this figure time point 2, that's after processed meat, and then also the phyto enriched meat, and you see that goblet cells and stem cells and transient cells are changing in time. But I was wondering, you also measured at baseline. What happens at baseline, and why is that not included in this figure?
So the baseline for the participants was their general baseline. It wasn't necessarily a run-in baseline. And if I remember correctly from this figure, I think this is the change from that time point.
So it's compared to the baseline?
I think so, yes. Meat.
Okay, because I was wondering, wouldn't you expect your intervention with the phyto meat to go back to baseline? If your processed meat would have a specific effect, or is that maybe not the effect that you would have?
Yeah, it's a good question. Not necessarily, because the baseline in this study, in the phytome study, wasn't controlled in a particular way where we had them all consume a certain type of diet, so then we can be sure that the different groups that we have, because when you take a collection of people, they all eat differently. So that's why in the Myblin study, we had that run-in phase to make sure everyone's consuming the right amount. So we don't actually have historical data from those participants, what they were eating like. They just collected a baseline. And then they added the processed red meat intervention, and then there was a white meat intervention, and then there was a processed red meat with the added phytochemicals. So I found comparing to this baseline less useful because for me it didn't represent a particular, an easier to confirm difference. It just tells you what were people eating before, and that can be very variable.
Makes sense then.
Yeah.
Okay. So you have these, you identified the cell types, then you looked at the differentially expressed genes, comparing time point 4 with 2, right? That's always your comparison. And then you performed an analysis to get a protein-protein interaction network. You used string for that. And I couldn't see in the methods what type of confidence score that you use to get the protein-protein interactions. Do you
Can you
know?
repeat that word? I didn't hear you.
The confidence score for the protein-protein interaction networks.
You're asking if I remember this score?
Yeah, do you know if was it strengthened or not? Because there's a lot of because I was missing also the complete network. Let's say you show the hub genes, which you nicely retrieved from it. But how was the complete network looking? The protein-protein interaction network.
From what I recall, these were the top 10 hub genes. So this doesn't show the incomplete network that we received, but we just isolated what would be the most important, assuming that because of that level of interactions that you expect between these genes and the expression that we see in those genes. So yeah, we didn't include all of the data there, but just what we thought would be the most relevant, because this is already maybe kind of complex on its own.
But why is protein-protein interaction then relevant biologically?
So this was done in order to kind of build more hypotheses. So we were curious if we see these different genes being expressed, can we kind of get a sense or an idea of what this might mean to the protein side? Gene expression doesn't necessarily mean proteins are being made this way, because there's steps in between that could affect this. But this was more of hypothesis building and just trying to get a sense of what this could mean. That was the goal of this chapter, was to see, is it possible, given a gene transcription, if you have gene transcription data from bulk tissue, can you, and you don't see very big differences between the groups, because often that happens with lifestyle interventions, can we get down to the cell level and potentially see changes that might be more relevant, that are kind of blocked by the overall averages, when all the expressions are averaged into one number?
Yeah, I agree with you there. I think that's an elegant way to do this. But indeed, looking at your protein-protein interaction and then only identifying the hub genes, that biological meaning is a bit missing in my opinion. But, yeah.
Mrs. DeBenedictis, as you hear and see, the time for defending your thesis has passed. The committee will now withdraw to discuss the quality of your thesis, and in particular the quality of your defense, and please await our return.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Hit the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
So everyone is sitting.
Yep.
Julia DeBenedictis. The degree committee here present has assessed the quality of your thesis and the quality of your defense. And in view of its positive verdict and taking into account your previous qualifications, the committee has decided to grant you the degree of doctor. Professor de Kok is authorized to confer upon you this academic distinction in accordance with Dutch university custom and law. And I invite now your supervisor to take the floor.
Dear candidate, because you're still a candidate, do you promise to work in accordance with the principles of scientific integrity at all times, to be careful, honest, transparent, independent, and responsible?
Thank you. Yes, I promise.
That's the right answer. By the authority vested in us by law and conformity with the decision of the committee here present, I hereby confer upon you, Julia Nicole DeBenedictis, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and all other members of the committee and affixed with the official seal of Maastricht University. Congratulations. Dear Dr. DeBenedictis, before I hand the microphone to your supervisor, your daily supervisor, Dr. Van Breda, to give the laudatio, I really have to say that it was really my pleasure to be your supervisor in this trajectory that we have been in for such a long time and interrupted by COVID also. And I was impressed really by the speed in which you accommodated all the skills that were new to you, how to handle all the different samples, data set, and manage all of this. That was really a privilege to see and be part of. So I'm convinced that you, together with Frank, are looking forward to a brilliant future, and I wish the two of you all the best. And now I give the floor to Simone.
Dear Dr. DeBenedictis, dear Julia, I'm truly honored to be the next one in congratulating you on your well-deserved doctoral title today. The end result is absolutely impressive and something to be very proud of. It has already been 6 years since we first met online together with Theo for your job interview. From the very first moment, you stood out above all the other candidates. We invited you to Maastricht for a follow-up meeting, during which you had to present your interpretation of one of our published human intervention studies. Your performance exceeded our expectations, and right then, Theo and I knew you were the right fit. We offered you the position on the spot. In our conversation, we asked what you believed to be the most important quality in a PhD candidate. You answered, taking ownership. And that is precisely what you demonstrated throughout the entire project, even at times when faced with significant challenges. Because not only the research topic itself was about navigating complexities, also some circumstances you encountered during your PhD journey were rather complex and requested both navigation and ownership. The first major challenge came in March 2020, just after the first year of participant inclusion in the MyBlend study, when the COVID-19 pandemic hit. From one day to the next, the study had to be paused, and participants were unable to complete their interventions. What followed was a period of uncertainty, it was unclear how long the disruption would last and what the impact on your project would be. However, you seized this period as an opportunity to learn bioinformatic techniques, applying them to an existing data set to generate relevant data for your PhD project. Meanwhile, you developed a safety protocol to resume the MyBlend study under strictly controlled conditions. Although the pandemic's impact lingered throughout the study, your dedication and effort ensured the inclusion of the anticipated number of participants. In addition to the complexities due to COVID, you had to deal with several complexities of personal nature. Just to name a few. Being separated from Frank during weekdays when he began working in Leiden. Dealing with your chronic condition that caused persistent joint pain and fatigue. Moving 4 times in 4 years. And perhaps the most unexpected and difficult event. The sudden heart attack from your father. The emotional complexity of being thousands of miles away from your family during such a critical time was tremendous. Again, an uncertain period followed, and this one could not be tackled with any protocol or new technique. Yet you demonstrated a different kind of ownership. Ownership of the needs of your father, your family, and yourself. You immediately flew back to Texas to support them in every way you could. Despite this, you remained committed to your research, processing results, analyzing data, and working on manuscripts. After a few weeks, you returned to Maastricht, but soon had to go back as it became evident that your father had little time left. After his passing, it wasn't easy to pick up where you had left, but you did, knowing it was what your father would have wanted. Despite how hard it was emotionally, you continued, finalizing your chapters one by one. Some of your work has already been published, and now you have successfully completed and defended your thesis. You have since begun a new chapter in Leiden, living there together with Frank, and starting your role as a nutrition scientist at Danone, where your aim is to translate complex research into actionable insights that can truly impact public health. My hope is that any complexities in your life remain in your research, while your personal life is as simple or difficult as you choose it to be. Because after all, simplicity is often the most complicated skill. Finally, I want to express again my congratulations on earning your doctoral title. I would also like to acknowledge your partner Frank, your mother and stepfather, who unfortunately cannot be here today due to surgery of your stepfather, your stepmother and stepsister present here with us, your twin sister Jennifer, and all your other siblings, I lost counting, your parents-in-law, other family, friends who have supported you along this journey. I wish you continued success in both your scientific career and personal life. Enjoy this very special day, today and for the rest of your life.
Thank you.
Please sit down. Esteemed Dr. DeBenedictis, dear Julia, I would like to congratulate you on the bottom of my heart for having this doctorate at Maastricht University. And I congratulate also you on behalf of Maastricht University. And I would like to share some impressions of the committee with you. First of all, we have seen a clear, but also a very extensive and elaborate explanation of your work and in your summary, the presentation that you started with. And I hope that it is clear to all people that are not so familiar with the work that you have done. Your thesis is considered as very good. And it's special in terms of its diversity of the nutrient analyses, the models that have been used and the method that have been applied. And also it's special in terms of the strict design of the structure. The thesis is multidisciplinary, comprehensive, and it shows also an impressive amount of scientific work. And also, I've seen that it's also well published already, and lots of work is still underway to be published. Your defense, it was very good. You listened very well. You were in your answers philosophical, elaborate, creative, and you have very well shown the scientific to be very knowledgeable of the scientific background of your work. And the committee is very satisfied with the quality of your defense. I do not only congratulate you with that, but also your two supervisors, Professor Theodor Kok and Dr. Simone van Breda. Congratulations with the results that you have seen in the last 5, 6 years. A long time, and you have seen also an excellent PhD candidate working on in your laboratory. So congratulations with all the results that we have seen today and earlier. And of course, I would like to share my congratulations with your elaborate family. And I would like to congratulate you with a fantastic well, daughter, sister, and so on, and so on. So congratulations, and I would also include in my congratulations, I think a lot of colleagues and friends that are here present in the aula, but also have seen your presentation and your defense in the live stream. So again, congratulations with that. Thank you, all members of the degree committee and also of the thesis committee. I would like to thank you for critically reading the manuscript and also by having interesting questions and an interesting discussion today. So thank you very much, and in particular, the external members of the committee. Thank you very much. Maastricht University appreciates your contributions. Before I close this ceremony, I would like to invite your close family and your both paranymphs to go with us to take a picture on the stairs in the hall. And then I would like to, after that, I would like to invite all the people present here to go to the reception in the rafter, and you find it easily if you go out of this aula and you take a right turn after the elevator, you can get there easily. Hereby, I close this academic ceremony.
Take my book.